Hello, hello. Today's lecture is going to be on fetal development and genetics. So let's get started. We can break the stages of fetal development into three main stages. The pre-embryonic stage, which occurs from fertilization to about the second week. Keep in mind that at fertilization, all the genetic material is present because you have both the genetic material from the mom and from the dad. So the genetic makeup is determined at that moment of fertilization. The cells are going to continue to divide and develop into the blastocyst and the trophoblast. And then implantation takes place during this stage as well. So that is the pre-embryonic stage. Then we take a look at the embryonic stage, which is from weeks 2 through 8. And this is a stage of rapid development. By the end of the embryonic stage, all major organs are present. In fact, the primitive heart starts to beat as early as 3 weeks gestation. All of the main external features are also present by the end of the embryonic stage, including the arms, the legs, the fingers, the toes, and even the nose. The fetal stage starts at week 9 and lasts until the delivery of the baby. During this stage, all of the organs and structures are growing and developing. The embryo initially is composed of 3 layers of tissue. Each layer serves a specific function in development. The endoderm layer, which you see here, depicted in yellow, becomes the digestive system, the liver, the pancreas, and the inner layers of the lungs. The mesoderm layer, which you see here in red, becomes the circulatory system, the epithelial layers of the lungs, and the skeletal system. It also becomes the muscular system. The ectoderm layer, which you see here in blue, will become the hair, the nails, the skin, and the nervous system. You can see a few other structures here that will change and become important in the fetal stage as well. We're going to look at the amniotic cavity with the amniotic fluid. We're going to take a look at the amnion and the chorion. You can also note that the yolk sac, which is right here, is what provides some nutrients to the baby before the placenta is fully functioning. Teratogens are agents that can impact the normal development of the baby. The embryonic stage, when there is rapid cell division occurring, is when the embryo is most susceptible to the damage from outside sources, and therefore when teratogens can have the most drastic impact on the development of the baby. Teratogens can be environmental, such as exposure to radiation, infectious agents such as TORCH, which stands for toxoplasmosis, other agents, rubella, which is also known as German measles. The C is for cytomegalovirus, and the H for herpes simplex. Those are all infectious agents that can have a drastic impact on the development of the embryo. We also look at chemicals such as drugs that can impact the growth of the embryo. For instance, there was a drug that was widely used in the 50s, I believe it was, that for severe nausea with pregnancy, so used for morning sickness. And it was discovered that this drug led to physical malformations of the baby. Street drugs will also cause harm to the baby, and there's some other prescription drugs that we just need to be very cautious about. There's also maternal conditions that can impact the growth and development of that baby, such as diabetes. So when we have out of control maternal diabetes, we have a embryo and a fetus that's constantly bathed in incredibly high levels of blood glucose. And this can lead to all sorts of complications, including some malformations of the heart and congenital heart diseases. Go ahead and jot down some of your key takeaways from fetal development. So there are three main stages of fetal development: pre-embryonic, embryonic, and fetal. All genetic material is present at the moment of fertilization, and all major organs are present at the end of the embryonic stage. And there are three embryonic layers that develop into all bodily structures: the ectoderm, mesoderm, and endoderm. And then we want to keep in mind that teratogens can impact the development of the fetus, and a key teratogen to remember is maternal conditions such as diabetes. The magical placenta. The placenta is the only organ that can be regrown as many times as it's needed, and it's a disposable organ as well. We throw it away when we're done with it. The placenta provides oxygen and nutrients to the baby and removes waste products from the baby's blood. The placenta has a fetal side, which you see right here. So this smooth side is the fetal side. What you're looking at is that amnion layer, the amniotic sac. And if we were to pull it up from here, we would see where the baby lived. So you can see the umbilical cord came out and attached to the baby. And a maternal side. So this is the side that was attached to mom. And doesn't it? It looks like it would be attached to a to the muscle there, right? Or to the uterine wall. The placenta acts as a transfer organ and as an endocrine organ. And the functional unit of a placenta is called a cotyledon. And you can see the cotyledons here. These are the functional units of the placenta where the exchange occurs. As you can see, there isn't a mix of the fetal and the maternal blood. The maternal blood supply feeds the placenta, but does not go directly to the fetus. So again, if we look at this upper picture here, we have our maternal blood supply here, which enters and feeds those cotyledons, but then the fetal blood supply comes in and it does the exchange. So there's not a direct link between the maternal blood supply and the fetal blood supply. The placenta is also responsible for producing the hormones to sustain the pregnancy. The placenta creates a barrier similar to the blood-brain barrier, so that not everything in the maternal blood will pass through the placenta. For instance, insulin cannot cross the placenta, but glucose can. Certain drugs can't cross the placenta, and luckily some infectious agents can't cross the placenta as well. The hormones produced by the placenta help to sustain the pregnancy and are HCG, human placental lactogen, which is considered HPL, estrogen, progesterone, and relaxin. Relaxin actually allows the mom's body to change and grow to accommodate the growing fetus. So it does what its name indicates. It relaxes the tendons and the ligaments and the muscles so that the body can grow. Due to relaxin, mom is at a higher risk of musculoskeletal injuries during pregnancy. It also leads to runny noses. So mom might have an increase in mucus secretion due to the relaxin production. So the picture here is of a 10-week-old fetus surrounded by its amniotic fluid. The amniotic fluid is contained in the amniotic sac, which, remember when I pointed out the amnion, when we looked at those layers of the embryo, the embryonic tissue layers, the amnion creates the amniotic sac. The amniotic fluid serves many purposes for the growing fetus. It helps to maintain fetal body temperature. It allows for symmetric growth because it creates space around the baby and prevents there from being pressure on one particular area. The amniotic fluid helps protect the fetus from trauma as well by absorbing any potential blows. Similarly, it provides protection for the umbilical cord by allowing space for the cord so it doesn't become compressed. Another purpose of the amniotic fluid is to allow the free movement of the fetus, which aids in the development of the musculoskeletal system. A little later in its fetal journey, the baby will begin to drink the amniotic fluid, and the kidneys will begin to function by filtering it out, and the baby will pee it out and drink it again. So it's a nice little cycle. The amount of amniotic fluid can give us an indication of the health and well-being of not only the baby, but also of the placenta, because the placenta contributes to the development of the fluid. But you can see pretty clearly here how well protected that baby is in that space. The umbilical cord forms from the amnion layer as well, and a normal umbilical cord is made up of 2 arteries and 1 vein. A good way to remember this is to think of Ava. So we're going to call our umbilical cord Ava because there are 2 small arteries and 1 large vein. Remember, the definition of an artery is a vessel that takes blood from the heart, and a vein is one that returns blood to the heart. So although we commonly think of arteries carrying oxygenated blood and veins deoxygenated blood, we have to keep in mind that the definition when it comes to the fetus. The vein is what carries the oxygenated blood from the placenta to the fetal heart, where it is then pumped into circulation. And the arteries carry the deoxygenated blood from the heart to the placenta for it to be processed. Wharton's jelly is a connective tissue that protects the vessels within the cord from being compressed. You can see here the thickness of the cord, and you can see kind of the vessels within the cord here. These dark lines are the vessels within the cord. And so the whiteness that you see is all the Wharton's jelly that is within the cord. And if we go back to our magical placenta slide, you can see this as well. So we have the cut cord where you can see some of the blood, and we have the Wharton's jelly, which is the connective tissue that protects those vessels within the cord, because those vessels don't have a lot of structure to them, right? Just like our veins and arteries. And our veins and arteries are protected by our musculoskeletal system, but the umbilical cord doesn't have that. So it's the Wharton's jelly provides that protection. And we'll talk more about some of the abnormalities that can occur with the cord and the placenta and how those can impact the pregnancy when we talk about pregnancy complications. So if the umbilical cord carries blood to and from the placenta, and the placenta acts as a transfer organ and endocrine organ, what is going on with that baby's lungs and their liver? Well, the liver is functioning at a very limited capacity in the fetus. And in fact, babies are born with an immature liver. remember this when we talk about the newborn medications that we give in the newborn lecture. The lungs are filled with amniotic fluid, so the fetus isn't breathing. To accommodate the fetal environment, there are a few adaptations in the fetal circulation. The three main adaptations are the three shunts. We have the ductus venosus, which bypasses the liver, the ductus arteriosus, which bypasses the lungs, the foramen ovale, which connects the right and left atrium. So let's go a little bit further into fetal circulation. So we're going to take a look at the blood flow in fetal circulation. The blood that flows through the fetus is actually more complicated than after the baby is born. This is because the mother, or the placenta more specifically, is doing the work that the baby's lungs will do after birth. The placenta accepts the blood without oxygen from the fetus through the vessels that leave the fetus through the umbilical cord, which are the umbilical arteries. Remember, there are two of them. Ava. When blood goes through the placenta, it picks up the oxygen, and that oxygen-rich blood is then returned to the fetus via the third vessel, the umbilical vein. The oxygen-rich blood that enters the fetus is sent through the ductus venosus, the shunt that passes highly oxygenated blood through the liver to the inferior vena cava, and then to the right atrium of the heart. A small amount of this blood does go directly to the liver to give it the oxygen and nutrients it needs to grow. The oxygen-rich blood goes through one of two extra connections in the fetal heart that will close after the baby is born. There's your ductus venosus. Sorry, I forgot I had that in there. So we have the next one, which is the foramen ovale. And this is the hole between the top two heart chambers, the right and the left atriums. And this is going to allow the oxygen-rich blood to go from the right atrium to the left atrium, and then to the left ventricle and out the aorta. As a result, the blood with the most oxygen gets to the brain, and this also allows for the initial bypass of the lungs. Blood coming back from the fetus's body also enters the right atrium, but the fetus is able to send this oxygen-poor blood from the right atrium to the right ventricle, the chamber that normally pumps blood to the lungs. Most of the blood that leaves the right ventricle in the fetus bypasses the lungs through the second of the two extra fetal connections, or the fetal shunts, which is right here, and that is the ductus arteriosus. So the ductus arteriosus is going to send the oxygen-poor blood to the organs in the lower half of the fetal body. This also allows for the oxygen-poor blood to leave the fetus through the umbilical arteries and get back to the placenta to pick up its oxygen. These two bypass pathways in fetal circulation make it possible for most fetuses to survive pregnancy even when there are complex heart problems and not be affected by them until after birth. And that usually it's when these pathways begin to close that they start to have some issues. So keep in mind your three shunts: the ductus venosus to bypass the liver, the foramen ovale, which allows the oxygen-rich blood to go to the head or to the brain, and the ductus arteriosus, which is the primary bypass of the lungs. So what are your key takeaways for the growing fetus? So the placenta acts as lungs and a GI tract for the fetus, allowing for gas and nutrient exchange. The umbilical cord, AVA, is the lifeline from which the placenta to the fetus, the lifeline from the placenta to the fetus, and is protected by Wharton's jelly. Amniotic fluid plays a role in protecting the baby. And there are three main shunts to bypass the liver and the lungs: the ductus venosus, ductus arteriosus, and the foramen ovale. So now we're going to take a look at genetics. Genetics is the study of heredity and its variation. Genes are the individual units that make up DNA and are responsible for heredity and for the traits that each individual expresses, such as having green eyes. A chromosome is a strand of DNA that carries genetic information. Humans have 22 sets of chromosomes in addition to the sex chromosomes. And a karyotype is a pictorial analysis of the chromosomes. So let's look at this a little bit closer. Here is an example of a karyotype. As you can see, it is a picture of an individual's chromosomes. We can see here the 22 sets and the sex chromosomes. Do you think this is a male or a female? Give you a hint. Here's our clue right here. We have two X chromosomes, so this is the karyotype of a female. Karyotypes are commonly obtained from white blood cells or from fetal cells. The fetal cells can be obtained one of two ways. An amniocentesis can be done, which is a procedure performed by a physician, and it uses a large needle to withdraw some of the amniotic fluid while the baby is still in utero. There's fetal genetic material contained within the amniotic fluid, so we can then isolate that genetic material to view a karyotype. The other method for determining a fetal karyotype is by isolating a small amount. There's a very small amount of fetal genetic material that enters maternal bloodstream. And this method was introduced about 10 years ago. So that's kind of something kind of cool to think about, is that there is genetic material of every single pregnancy within the maternal circulation. So mom carries on to her kids, no matter how early or late that baby may have passed, or later in life, if they unfortunately lose that child, they're still holding on to that genetic material. So from the point of implantation on, the mom will have some of the genetic material in her blood circulation. Some of the reasons we do this is to determine if there are any chromosomal abnormalities present. Sometimes those are good to know before birth so that we can prepare the family and so that we can be prepared at birth. Karyotypes are organized by size. We have the largest set of chromosomes as number 1, and the smallest is number 22. And then the sex chromosomes are designated as X and Y. Everyone has at least one X chromosome, and females have 2 X chromosomes, and males have 1 X and 1 Y. This is important to keep in mind as we talk about some of our patterns of inheritance and problems that come along with the patterns of inheritance. So patterns of inheritance demonstrate how genetic disorders can be passed on to a person's offspring. A genetic disorder is a disease caused by an abnormality in an individual's genetic material or genome. Mendelian, named after Dr. Mendel, who used peas to study genetics and monogenic disorders, are disorders caused by variations in a single gene and are typically recognized by their striking familial inheritance patterns. And these are going to be our autosomal dominant, autosomal recessive, and X-linked inherited diseases. Sickle cell anemia and cystic fibrosis are some of the common autosomal recessive diseases that we think about. So let's take a closer look at each of these types of inheritance. Here is a representation of autosomal dominant inheritance. Dominant genes will be depicted as uppercase letters and recessive as lowercase. As you can see, it only takes one gene to pass along the trait. Another way to think about it is as a child who has one parent with the mutated gene has a 50% chance of inheriting that mutated gene. Keep in mind, each pregnancy is a unique event. So if child 1 bears the gene, doesn't mean that child 2 won't. Child 2 also has a 50% chance of inheriting that mutated gene. Where with dominant genes, only one has to have the gene to express that gene. With recessive genes, both must have it to express the trait. Those that only have one are carriers, but don't express the trait. As depicted here, 2 unaffected people who each carry one copy of the mutated gene for an autosomal recessive disorder have a 25% chance with each pregnancy of having a child affected by the disorder. Now, what if mom in this picture wasn't a carrier? Do you know what the chance would be that her offspring would express the trait? So if mom's not a carrier and dad is a carrier, there's actually a 0% chance, because remember, they would need both recessive genes to express the trait. Again, sickle fibrosis, I'm trying to say them both at once. Cystic fibrosis and sickle cell anemia are both examples of autosomal recessive disorders. We will get more into the specifics of those diseases in pediatrics. To understand the concept of X-linked inheritance, we have to think back to our karyotype. Remember that males have one X chromosome and one Y chromosome, and females have 2 X chromosomes. Males can only get an X chromosome from their mother, while females get an X chromosome from both parents. As a result, females tend to show higher prevalence of X-linked dominant disorders because they have more of a chance to inherit a faulty X chromosome. X-linked dominant disorders are generally pretty rare. For the same reasons that females are more likely to have X-linked dominant disorders, X-linked recessive disorders rarely affect female offspring. Males are impacted by X-linked recessive disorders because they only have one X chromosome. Therefore, if that chromosome contains the disorder, it will be expressed. Red-green colorblindness and hemophilia are the most commonly known X-linked recessive disorders. So looking back at our patterns of inheritance, there's also multifactorial diseases, which are caused by a combination of the effects of multiple genes or by interactions between genes and the environment. Some examples are Alzheimer's disease, heart disease, and hypertension. We also have non-traditional inheritance, and this does not follow the classical Mendelian laws regarding equal contribution from both parents. We don't get too much into these types of inheritance, but just know the definition of them. In addition to disorders that are inherited, we can also have chromosomal abnormalities. There can be an abnormality in the number of chromosomes, for instance, trisomy 21, which is depicted here. You can see the extra chromosome on 21 here, is the cause of over 90% of Down syndrome cases. There's also trisomy 18 and trisomy 13, and although they are rare, they are the second and third most diagnosed trisomies. Both are considered somewhat incompatible with life, and although there are cases of individuals with these chromosomal abnormalities living for a few years or even beyond, most will pass within the first year if they survive the birth. In addition to abnormalities in number, we can see abnormalities in structure. Chromosome abnormalities of structure usually occur when there is a breakage and loss of a portion of one or more chromosomes, and during the repair process, the broken ends are rejoined incorrectly. These can be deletions, duplications, and inversions, which you can see depicted here. Sex chromosome abnormalities can occur as well. Sex chromosome abnormalities occur when a person is missing a whole sex chromosome or has an extra sex chromosome. Abnormalities can also occur when a person is missing part of a sex chromosome. Syndromes that are caused by sex chromosome abnormalities are less severe than those abnormalities on chromosomes 1 through 22. For instance, Turner syndrome is caused by the deletion of part or all of one of the two X chromosomes. Girls with the syndrome are typically short and with loose skin on the back of the neck. They have learning disabilities and an inability to undergo puberty on their own. Treatment with hormones can stimulate growth and initiate puberty. Whereas on the other hand, fetuses who are missing a non-sex chromosome won't survive. So what do you think are some of the nursing roles and responsibilities along the lines of fetal development and genetics? I hope you've taken the chance to write down the questions that you have during this lecture, and we will discuss them at our next class meeting, or you can plop them into the student forum and I will check that and make sure to answer your questions. I hope you have a great rest of your day.